So this gives you a nice detailed view of the inside of a mitochondria. We see right here the matrix, this inner area, closed off by this membrane here, and then the outer, the cytosol. Where the glycolysis is happening, for example. And these are all the various proton pumps that are used. They're called ATP synthase. Protons are being pumped here, and then all kind of getting crowded and collected, high concentration, and then coming back through here, making the ATP. So this is where this chemiosmosis that I was talking about is happening. Just the movement of these hydrogen ions back and forth through the membrane. And it's driven by, the energy to do this is driven by the NADH, the FDH, the FADH2 that, remember, we made during the Krebs cycle. So from the Krebs cycle, we get these molecules. And NADH, when it's used up, it turns into NAD+. It'll go back and get recharged later on. Remember, where is the Krebs cycle taking place? Remember, in the matrix. Say it with me, matrix. And then you see all these hydrogens being pumped into the intermembrane space. Remember, this is low, remember, I'm telling you, it's low volume. So it gets filled up pretty quickly, and that makes it want to come back, as we'll see in the next few slides. And these electrons just get passed through these membranes. These are different membrane proteins used in respiration. A lot of these proteins, remember, proteins come from genes, specific genes in the DNA, and you have protein synthesis occurring. The mitochondria has its own DNA, where it has the DNA code to make these proteins that are stuck in this inner membrane. And after these electrons, they're going through the chain, they end up powering something very important. Remember, one of the products of respiration we have right here is water, and this is, now we can see where that water is produced. The electron drives with the, and remember, we need to breathe oxygen, so the oxygen, those electrons combine to form water. So we see where that oxygen is used, why you need to breathe. You need to breathe because this oxygen is the final electron acceptor. Probably gotta go back to red. Final electron. That's why you need to breathe. To power this process along, this chemiosmosis, to pump these ions, you need to keep breathing. And this kind of gives you a fun thing, I think. So NADH, FADH2, they're saying, we're sick and tired of carrying these electrons around. And over here, this is oxygen saying, wow, I love them. Give them to me. So that's what happens. These electrons go through these proteins like we just saw. As it happens, pumping those hydrogens, then ends up adding driving that process of creating water. Okay, that's good. That's why you need to breathe. You got to power this energy producing process in your mitochondria. Final electron acceptor. So this is about chemiosmosis, protons diffusing back through this inner membrane to make ATP. We see the ADP well, actually, I messed up a little bit there. This is meant to show these hydrogens going from the intermembrane space into the matrix. As it does that, it powers this regeneration of ADP back into ATP with the triphosphates right here. So what we talked about, the semi-permeable membrane, this inner membrane, this is the ATP synthase, this blue arrow. Put that back there. Synthase. And these ions, hydrogen, that hydrogen, being a type of proton. So I'll put that here, proton. They just flow through there and they help to regenerate that ATP. So that's that chemiosmosis component. And like we mentioned before, that oxygen is the final electron acceptor. Get water out of it, so the oxygen combining with those hydrogens is going to create that water. Oxidative phosphorylation, electron transport chain, chemiosmosis. That's what we got. It's a beautiful thing. So here we have these are the cristae. Sorry, that's the matrix. The outer membrane, the matrix to the very inner side. These little kind of finger-like things. Those are those cristae, that inner membrane, like we just saw in chemiosmosis. That's where that chemiosmosis is working. Matrix, that's where our Krebs cycle is working. And 70S ribosomes. Now remember, this is small surface area. So structurally, that's what makes it well adapted for this chemiosmosis. Not small volume. That means a high concentration of ions builds up quickly. Don't have to wait too long to get those hydrogen ions flowing back through the ATP synthase. Look at all these folds, many folds. That equals high surface area, lots of space for chemiosmosis and the electron transport chain, all those proteins in there. So we have a lot of these electron transport chains happening at once. So lots of electron transport and chemiosmosis happening. And I like that. Those are the main ones. And electron tomography, being able to use the power of the electron microscope to really dive in and see the inner workings of the mitochondria, showing that the cristae are connected. They do show it connected to the intermembrane space and the shape and volume. Look at how much this is changing. Show the shape changing over time. So 3D active. So they're not killing the mitochondria. That's interesting because usually you have to kill kill the organism to view it under an electron microscope. But here, they don't have to. You might have to, on the exam, look at a diagram and figure out whether, decarboxylation or oxidation reactions occur. So I gave you some information. You can look back like here, seeing that loss of carbon is showing you oxidation. Also seeing the loss of hydrogen. When you're building the molecule, when you're adding the hydrogen, you're doing reduction. And when you're pulling, remember you're right here, when you're losing that, that's oxidation. So your oxidation. So it just takes practice. I mean, to kind of figure that out. I gave you a couple diagrams here. So, like for example, two places where decarboxylation occurs, and this you might recognize. This is the Krebs cycle, and why we would select that. So basically, here we go from 6 carbons to 5. That's a decarboxylation because you're reducing the number of carbon atoms, which kind of is implied, in the name. So hopefully that'll be a good key to figure that out. And here's a repeat. I did have this twice here, showing this loss. Example 5 hydrogens down to 4 hydrogens. So we're losing hydrogen. Oxidation is loss. Oil rig, I think, is the easiest one, especially in Long Beach. A lot of oil rigs around here. Long Beach, a lot of oil rigs. But it's confusing, right? Because this is oxidation in here to these molecules that are shifting through the isocitrate, the ketoglutarate, succinyl-CoA, succinate, fumarate, malate, all those things. In that cycle, they're being oxidized, right? But these other molecules that we need to pay attention to, they're being reduced. So it's just, hopefully this great diagram, this great explanation, you'll be less confused. Here's another nice, almost looks like a heart. Seeing loss of this carbon, so decarboxylation. And also, if you see CO2 coming off of there, carbon, CO2 has carbon in it. So again, my nose itchy. Sorry about that. And then here being reduced. And the last thing, I need you to draw and label, annotate. I love annotation. A mitochondria. Make sure you pay attention to everything we've been talking about. So outer membrane. If you have an outer membrane, probably sure you have an inner membrane. And your cristae, those little finger-like folds, and the matrix. All right, and then go ahead and add, a little ribosome in there. Little DNA's in there. Remember, it used to be its own a long, long time ago. It was living on its own. Maybe 3 billion years ago. I think that's when, from what I remember, 3 billion, 2.5 billion years ago, the eukaryotic cell is thought to have started based on the research information. So go ahead and make a nice detailed one on this last slide.